So I want to finish off the discussion talking about why three-dimensional shape is so important for proteins. And I've already mentioned it, right? So I've already given you a basic sort of definition, but I want to illustrate that with specific examples, right? And we'll talk a little bit in this discussion about what proteins do as well, right? So the reason that three-dimensional shape is so important, as I've already mentioned, is just like it is for our body, the shape of a protein determines its function. Right? So remember our discussion about complementarity. Right? The shape of a protein determines its function. The needed function of a protein will determine its shape. Right? The two things are not separable. Right? They're both intertwined in this process. The shape of a protein tells you what it's going to do. What a protein needs to do will in part determine what its shape should be, right? And a perfect illustration of this is what we see in the proteins in connective tissues, right? So I believe that you've started tissues this week in labs, so hopefully these terms will not be completely foreign to you, but right, connective tissues are bone, cartilage, tendons, fat, right? So all of these things that help to build and hold our body together, right? But right, as it says here, right, these tissues have widely differing properties, right? Bone is very different than cartilage, right? And cartilage is very different than fat, right? Bone is very rigid and very strong. Fat is very soft and malleable, right? Cartilage is stretchy, right? So each of these different properties come from the different proteins that you find in each of these tissues. Right? So the property of each tissue depends primarily on two proteins, collagen and elastin, and how much the tissue has of each. Right? So tendons are very strong and very rope-like. Right? So they're going to have more of one, whereas cartilage, being much more stretchy, will have more of another. Right? And so collagen proteins are long and rope-like. Right? And they actually wrap around each other like the strands in a rope. Right? So here we can see, so schematically is showing three different collagen chains. Right? One in green, one in purple, one in red. Right? And they're spiral shaped, so they're helical. Right? So you can see that here on these. And these spirals wrap around each other into what we call a triple helix. Right? So this is a triple helix. Right? But the consequence of those strands wrapping around each other is it makes them very, very strong. Right? It provides collagen protein with greatly enhanced strength. So if you were to take a rope, right, so here's our rope, right, and tease out any one individual little strand of a rope, they're very small and they're very thin and they're pretty weak. You can break them relatively easily. But because there are dozens or hundreds of these strands all wrapped around each other in a rope, right, the rope strands together have a much greater strength than any individual strand would have by itself. Right? And so tissues with lots of collagen have much greater strength. And the tissues that have lots of collagen are bone and tendons and ligaments. Right, so the ligaments that hold your joints together, the tendons that attach your muscles to your bones, the bone itself, right, are full of collagen, right? Because collagen is a very strong protein that provides the tissues that contain it with lots of strength, with lots of ability to resist physical force. Elastin, on the other hand, as you can probably guess from the name, right, are stretchy. Right? And so elastin has a different structure, where the proteins are loosely attached to each other, not tightly wound. Right? So in this picture, each little green line, and I just covered it over with a red line, each little green line is a single elastin protein, a single elastin chain. Right? And they are connected to each other by these the red lines are chemical bonds that are called cross-links. Right? And so what this allows them to do, because they're loosely attached to each other, is it allows elastin to stretch and relax like a rubber band, right, which provides what we call elasticity or stretchiness to tissues. Right, and so we would expect to find more elastin in tissues that are stretchy. Right, so for example, cartilage. Right? The connective tissues that make up our skin, that areolar connective tissue, is a pretty even mix of both collagen and elastin. So skin is both strong and stretchy in equal measure. Right? It's not particularly good at any one of those things. Right? It's not super strong, it's not super stretchy, but it has a little bit of both. Right? It can be both strong and stretchy at the same time. Right. Another big key, though, for things that are not structural, right? Say, let's look inside of a cell, right? And the proteins that actually do some work in a cell, right? Most proteins need to be able to attach to other molecules to perform their function, right? So hemoglobin has to be able to attach to oxygen. Insulin has to be able to attach to a receptor on the cell it's trying to send a signal to. Lactase has to be able to attach to lactose in order to be able to break it down into glucose and galactose. Right? That attaching in the study of these molecules we call binding. In fact, I've used that word a couple of times already, right? Binding, to say that something binds means that it attaches. Right? And so proteins are built so that their 3D shape closely matches the 3D shape of the molecules to which they bind. In fact, the metaphor usually used to explain how this works is a lock and key. Right? That a key is shaped perfectly to open the lock it matches to. Right? Where the enzyme, the protein, is the lock, and the key is the thing to which it binds. Right? So switching that metaphor over to something that looks kind of like an enzyme. Right? So here we have an enzyme in green. It has what's called an active site, right? Which is where the molecule that it binds to will actually attach. Right? The substrate is the thing that it attaches to. Right? And you can see that that active site is perfectly shaped to allow that purple molecule to bind. Right? And if anything changes the three-dimensional shape, the ability to bind may change. And I'm purposely being vague about that, right? Because those changes can cause binding to increase. Right? It can make things bind more strongly. Or bind more weakly. Or even Not at all. Right? Either change is possible. Right? If the change that occurs in the protein makes the shape more like what it's trying to bind, if it makes it closer, a better match, right, then it may bind more strongly to that thing. Right? But if the change makes the 3D shape alter in a way that makes it more different than the things it's trying to bind, then it will attach to that thing more weakly. Right? Perfect example to pull something straight out of real life is with COVID. Right? So when we look at the virus itself, right, viruses mutate very rapidly. And so one of the big concerns now are with these new variants. Right? And one variant in the UK that comes from the UK is more transmissible, right? Which means that it spreads more quickly. And one of the big reasons that it spreads more quickly is that it acquired a mutation that changed the three-dimensional shape of its what's called spike protein, right? And the spike protein is what attaches to a receptor on our cell and allows it to get inside so that it can infect. And this mutation makes the spike attach more strongly to our receptor, right? It made the shape better for attaching to the receptor on our lung cells. And so that makes it able to get in and cause an infection at even lower amounts. That's why it's more transmissible. Right? Or on the flip side, there's a South African variant that has a mutation that causes antibodies to bind more poorly. It changes the place that the antibodies attach to on the virus, so that the antibodies can't bind as well. Right? It makes those antibodies less effective. Right? So both of these things are possible. Right? It's possible that a mutation can cause a change in a protein that results in it binding more strongly to something. Right? Or if it alters the 3D shape so that it's less like what it's trying to attach to, right? So that the shape becomes wrong, right? It will bind more weakly or potentially not at all. And let's look at some examples from the human body. Right? And typically we focus on things that make binding worse because they end up causing disease. Right? So in A&P and especially in medical physiology, we tend to focus on the things that make binding worse, right? Because those are the things that usually cause disease. Right? So the first thing is, let's look at an enzyme. Right? And I've used that term a couple times. Right? And you may be familiar with what an enzyme is. You may not be. An enzyme is a protein that performs a chemical reaction. Right? They speed up chemical reactions like a catalyst does. Right? So our prime example here that I've mentioned a couple times already is lactase. Right? It breaks down the disaccharide lactose into glucose and galactose. Right? It breaks that chemical bond between the two and separates them into the individual monosaccharides. Right? And in order to do that, lactose binds to the actual lactase protein. Right? And once bound, that the protein causes the chemical reaction that splits this bond so that galactose and glucose can move separately. Right, so the lactose molecule attaches to lactase, then the enzyme breaks the bond between them. Right, and if the 3D shape of lactase changed, right, we may not be able to bind lactose. And if you can't bind lactose, then you would be unable to metabolize it. And as I talked about briefly, there are some people who are unable to metabolize lactose, right? That actually happens for a different reason, right? The reason is that our body, in some people, their body just stops making lactase, right? So there's no mutation involved here for people who are lactose intolerant. Their body simply stops making the enzyme, right? But were a mutation to occur, right, let's say right here, right, that changed the shape of this protein, right, then the lactose would no longer be able to bind. And so you would lose the ability to metabolize lactose. Right. This happens in a disease called phenylketonuria, or PKU. Right, this is a genetic disease where you lose the ability to break down an amino acid called phenylalanine. Oh, did not want that to happen. Okay. Pen's misbehaving apparently. Sorry about that. Right. So normally we can break down phenylalanine with an enzyme called phenylalanine hydroxylase. Right. And so normally when we break it down, we follow this pathway, right, where the phenylalanine becomes some other useful chemicals, some carbon dioxide, and some water. Right, so if we have excess phenylalanine in our body, we simply break it down into other stuff that we can use. But for people who have PKU, they can't do that. The enzyme is broken. It's unable to bind phenylalanine. Right, the enzyme is changed so that it cannot bind phenylalanine, and so the enzyme cannot break it down. And so what happens is it gets converted into something called phenylpyruvic acid, right, which when it accumulates is toxic. Right, and it can lead to learning disabilities and stunted growth if not caught in time. Thankfully, it can be managed by dietary changes in many cases. In fact, if you ever look on the back of a diet soda, right, so Diet Pepsi or Diet Coke or whatever, it will say on it, there will be a warning on it for phenylketonurics that it contains phenylalanine. Right, so people who would, if you drink that diet soda, you'll accumulate this phenylalanine, and your body, their body can't break it down. And so they have to avoid things like diet sodas because they have phenylalanine in them. Right, and you can see that here on this label here. All right, so one more example here. Right? Our red blood cell's job, of course, is to carry oxygen. Right? And the protein in our red blood cells that carry oxygen is hemoglobin. Right? And the 3D shape of hemoglobin is required for proper binding of oxygen. And so remember, we had those 4 chains in hemoglobin, the 2 alphas and the 2 betas. Right? That three-dimensional shape is necessary for oxygen to be properly attaching and being carried by the hemoglobin to the rest of the body. Right? In something called sickle cell anemia, a mutation in one of the hemoglobin genes causes hemoglobin to lose its normal three-dimensional shape. Right, so here is normal hemoglobin, right, in a normal red blood cell, right, has this amino acid sequence in its beta chain. So valine, histidine, leucine, threonine, proline, glutamine, or glutamic acid, I should say. Right. In people with sickle cell anemia, their beta hemoglobin gene has a mutation to where that in the number 6 spot, that glutamic acid becomes a valine. And that single amino acid change is enough to make that hemoglobin start to behave strangely. Right? To where it causes the red blood cell to become a sickle shape. And these sickle-shaped cells cannot properly carry oxygen. They cannot properly absorb it in the lungs. They can't properly drop it off in the body. But also, that shape causes them to become lodged in blood vessels and lodged in other places where blood accumulates, like the spleen. And so it can cause damage to those vessels and damage to the spleen, which results in a lot of pain. So, last thing. In order for cells to receive signals from other cells, they have to have proteins called receptors. So we've used this term before when we talked about feedback control, right? That there's always something in a control system that acts as a receptor to receive a signal. Right? So we can use this term receptor in terms of talking about cells that receive signals, but we can also use it to talk about proteins on those cells that receive signals as well. Right? So ultimately it means the same thing in that we're talking about something that receives a signal from its environment. Right? But we can use this term to refer to the whole cell or just the protein on that cell that's receiving the signal. Right? And the way that receptors receive signals is by binding to those signals in most cases. Right? Especially if they're molecular signals. So for example, insulin. Right? Insulin has an insulin receptor on the cells that respond to insulin. Right? Things like pressure or temperature have different kinds of receptors, right? So they don't necessarily attach to a molecule. Right, but for those molecular signals, right, we have receptors that bind to those molecules to receive the signal. Right? And a big example here are the taste receptors in the tongue. And remember, we have salty, sweet, bitter, umami, etc. Right? So PTC here, which stands for something really long, phenylthiocarbamide, is a molecule that normally tastes bitter. Right? And we have in our taste cells bitter receptors. Bitter taste receptors. And you might ask yourself, God, why? Right? Why would we want to have bitter taste receptors? Well, bitter tastes are things that taste bitter are typically poisonous, right? So thousands upon thousands of years ago, when, before civilization, when we were just sort of out on our own, trying to discover what, we could eat and what we couldn't eat, things that were bitter typically were dangerous, were typically poisonous. Right? And so bitter taste receptors allowed us to distinguish things that might be dangerous. Right? Some people have a slightly different version of the bitter taste receptor with a slightly different shape, and so they can't taste PTC. For those of you that have had a biology class relatively recently, if you ever did those tasting papers, right, where you tried to test to see if you could taste the bitter taste, That's what's in the testing papers, is this molecule PTC. Right? And so some people genetically have a slightly different version that changes the receptor shape just enough that some people can taste it and some people can't, because the molecule can bind to some people's receptors but not others. Right. So the one thing that's potentially problematic about this protein folding, right, is that protein folding is not an irreversible or permanent change. Right? It can be reversed, and 3D shape can be lost. Right? And this loss of 3D shape is called denaturation. And it sounds like a really complicated word, but really it's just saying denature. Right? It's going away from its natural form. Right? So the D means away from. Right? So it's away from its natural form. And so because 3D shape is required for proper function, denatured proteins do not work. Right? And denaturation can be caused by mutation, right, genetic changes, or by environmental changes. Right? So things like acid, right, acid pH, or heat can cause denaturation of proteins. Right? In fact, it's the first important step in digestion of proteins in the stomach. Right? One of the reasons that we have acid in our stomach is to denature the proteins in the food that we eat. Right? Because causing those proteins to unfold makes them easier to break down and digest. And you can see this process for yourself if you ever fry an egg. Right, so this is applying heat to cause denaturation. Right, so egg proteins, the white is naturally clear. Right, and as it heats up, you can see it start to turn white. And that's because the proteins in there are losing their natural form. They're becoming unfolded. Right, this protein is called albumin. Right? So the protein will go through this phase of denaturation. And eventually when you cook it enough, they actually become physically attached to each other. Right? That's what this cross-linking is called. Right? So when the egg becomes solid, right, through cooking, it's those proteins becoming cross-linked. Right? So they're now all physically, or chemically, I should say, attached to each other. Right, so when you fry an egg, you're just accelerating this process of denaturation.